Okay guys, welcome to BTech Biology. Now in this video we're going to briefly describe the sliding filament model of how a muscle cell or a muscle fiber can actually contract and get shorter. And you guys will have to watch the video. You can pause it at any point and you'll need to make some additional notes within your student notes. So this diagram represents a muscle cell. And if you look in your student notes, you'll find this diagram or a diagram very similar to this. And we can see it's actually a very big cell. It's got lots of nuclei within it that sit just underneath the cell membrane called the sarcolemma. The cytoplasm is called the sarcoplasm, and within the sarcoplasm there's actually lots of mitochondria, and they're the site of aerobic respiration to make ATP. Now ATP is needed for the myofibrils to contract and get shorter. So the myofibrils are these cylinders in the sarcoplasm. And the myofibrils are made up of two types of filaments, thin filaments called actin and thicker filaments called myosin. Now, if we look at a myofibril in a bit more detail, we can see that it's actually made of repeating units called sarcomeres. Now, on our diagram at the bottom, if you have a look at the sarcomere, it's actually defined by what we call the Z discs or Z lines. So one Z disc or Z line to the next Z disc or Z line is a single sarcomere, and these will repeat all the way down the length of the myofibril. Now, the thicker purple lines are what we call the myosin, which is the thick filaments, and they form what we call the dark band. So the muscle cell, and therefore the myofibrils, have a banding pattern of dark bands and light bands. So we're going to look at this in a bit more detail next. So if you guys go into your student pack on page 16, you'll find this diagram at the bottom, and you must be able to label this. Now, the photograph at the top of the slide is an electron microscope image of a cross-section of a myofibril, one of those cylinders that we find inside the cytoplasm. So what you should be able to see is on the bottom, these lines in green are what we call the Z discs or the Z lines. Now each Z line will represent one sarcomere to the next Z line. So on our diagram at the bottom, we've actually got 3 sarcomeres. Now in the middle of each sarcomere is an M line, so M for middle, and the thick myosin filaments are actually attached to the M line in the middle of the sarcomere. The thinner lines are the thin actin filaments, and they're actually connected to the Z line, which is the boundary of the sarcomere. So hopefully you can see that the thin actin filaments overlap the thicker myosin filaments. Now, any place that you have the thick myosin filaments, we're going to call the dark band or the A band. Now you can remember that's a dark band because dark has the letter A in it, and we call it the A band sometimes. Now, the section of the myofibril in between the dark bands are called the light bands. And the word light has I in it, so sometimes it's also called the I band. So what you'll find down the length of the myofibril is that there's bands of dark, where you've got the myosin thick filaments, and then light bands, which is only the actin thin filaments. Now, if you guys go to this diagram in your student pack, what you'll find is the diagram represents a thin actin filament. So the diagram at the top is the thin actin filament, and you can see it's actually a protein, and it's made of these kind of white circles, which forms the thin filament. There are actually 2 other proteins that are attached to the thin actin filament. One is called tropomyosin, and the other one is called troponin. Now, tropomyosin is a very long protein. It's a bit like a big long thread. And what it does, it blocks binding sites on the thin actin filament for what we call myosin heads, which will be on the thicker myosin filaments. So tropomyosin is a big long protein that blocks the binding sites that would form cross bridges with the myosin heads on the thicker myosin filament. So So we can see tropomyosin is that big long protein there on the diagram that blocks the binding sites on the actin filament, and that prevents the myosin heads from binding to these particular binding sites. Now, calcium ions are involved in muscle contraction, and they're released from the sarcoplasmic reticulum. When calcium ions bind to the second troponin protein that's shown at the top, that alters the shape of troponin and it pulls the long tropomyosin out of the way, and that then exposes the binding sites on the thin actin filaments for the myosin heads on the thicker myosin filament. So the diagram at the bottom, you can actually see the big long tropomyosin protein has been moved out of the way. The black dots are the binding sites for the myosin heads on the thicker myosin filament that's not shown on the diagram, and they're now able to bind to these black dots to be able to form what we call cross bridges. So the black dots represent the binding sites for the myosin heads on the thicker myosin filament. Okay guys, so your activity now is to add some notes to the diagram on page 20. You can pause the video if you wish, step by step, and then just add the notes onto your diagram. You'll need to then condense these notes into a summary diagram in your red revision notebooks. So this is called the sliding filament model of muscle contraction, and it's a number of steps that shows the thin actin filament being pulled over the top of the thicker myosin filament. Now the thin actin filaments are shown in blue here on the top of each of the 4 pictures, and the long pink structure is the thicker myosin filament. Now you can see attached to the longer, thicker myosin filament is what we call a myosin head, and that's going to form a link or a cross bridge between the thick myosin and the thinner actin filament. So you guys have to learn this in a bit of detail, and you can start your description at any points on the cycle of events. I'm going to start over on the left hand side. So if you have a look at the myosin head that I've just circled in the pink circle on the left, hopefully you can see that there was a molecule called ATP that is bound to the myosin head. Now ATP is made from aerobic respiration inside the mitochondria of the muscle cell. And once it's bound, what then happens is the ATP gets hydrolyzed into ADP and phosphate. The ADP and the phosphate remain bound to the myosin head. So one of the roles of calcium in muscle contraction is to actually activate an enzyme called ATP hydrolase. And it's this enzyme that breaks down the ATP or hydrolyzes the ATP into ADP and PI, which causes the myosin head to go into a cocked or an active position, and this is called the recovery stroke. So these are key terms in yellow that you guys will need to add on to your picture and to learn. So calcium ions activate ATP hydrolase. That's an enzyme that breaks down or hydrolyzes ATP. The ADP and the PI, or the phosphates, are attached to the myosin head. The myosin head is now in an active position, okay, and that's called the recovery stroke. We also know that calcium ions can bind to troponin, which is a protein on the thin actin filament, and it causes troponin to change shape, and that pulls another longer protein called tropomyosin out of the way, and that exposes the binding sites on the thin actin filament to allow the myosin head to form a cross bridge. So there's a connection now between the myosin head of the myosin filament and the thin actin filament, and that's the diagram at the top of your page. So the myosin head has formed a cross bridge with the binding sites on the thin actin filament. Now what happens is the ADP and the phosphate are released, and that causes the myosin head to change shape back towards its original position. Now as the myosin head changes shape, it's going to pull the thin actin filament over the top of the thicker myosin filament. So there's a sliding effect of the actin over the top of the thicker myosin. That's why it's called the sliding filament model. Now this process is called the power stroke, which is where the actin filament is pulled over the top of the thicker myosin. And what you'll see if you look at the sarcomeres down the length of the myofibril, the Z lines actually get closer together because the actin's going over the top of the thicker myosin towards the M line of the sarcomere, towards the middle of the sarcomere. Now, once we've had the power stroke, what happens is a new molecule of ATP is going to bind to the myosin head, and that breaks the cross bridge between the myosin head and the thinner actin filaments. So the myosin head is released from the binding site, and that's represented on the bottom of the diagram. So every new ATP that combines to the myosin head causes the cross bridge to break. The myosin head is then released from the actin filaments, so the cross bridge is broken. And what will happen now is that the myosin head goes back into its original position, and then the whole cycle can repeat to allow that actin to be completely pulled over the top of the thicker myosin. So we've now got to the bottom of this diagram. If we then go to the left hand side once again, we can see that if there's enough calcium ions, the ATP that's bound to the myosin head can again be hydrolyzed to ADP and phosphate. That causes the myosin head on the left hand side of our diagram to go back into the active position. That's called the recovery stroke, and then it can form a cross bridge at the top of the diagram, again with the binding site on the thin actin, and the whole process keeps repeating until we've got complete contraction of the myofibril. Okay, so on page 20 you've got a little activity to fill in. So if you want to have a go at this yourself, and then you can obviously check the answers. There is a little bit of a typo on the activity. I've just crossed that out in red. So pause the video, have a quick go, and then check your answers. Okay, so if you want to go through the answers then, the first missing word was calcium ions. Now you can put the symbol Ca2 plus ions, or you can say calcium ions are released from the sarcoplasmic reticulum. Now the sarcoplasmic reticulum is a type of reticulum that surrounds each myofibril within the muscle cell. So it's a store of calcium ions, but it also releases calcium ions when muscle contraction is about to happen. The calcium ions then diffuse and bind to a protein called troponin, which alters the tertiary shape or the shape of troponin, and it causes the other longer tropomyosin protein to be moved out of the way. And that's really important because that then exposes the binding sites on the thin actin filament, which are going to allow cross bridges to then occur between the myosin heads on the thicker myosin filament. So the myosin heads bind to the binding site on the actin, forming a cross bridge. The myosin head then bends, which pulls the thinner actin filament a short distance over the top of the thicker myosin filament. This is called the power stroke. So the force that is put on the myosin head against the thin actin causes the actin to move, called the power stroke. The ADP and the phosphate that are still attached to the myosin head at this point then get released from the myosin head, and a new molecule of ATP can bind to the myosin head. Now when the new molecule of ATP binds to the myosin head, that causes the cross bridge to break. ATP is then hydrolyzed to ADP and phosphate by the enzyme called ATP hydrolase. Now the second role of calcium ions is to bind to ATP hydrolase and activate that enzyme. So calcium ions have two roles to play. One is to activate ATP hydrolase, which breaks down and hydrolyzes ATP to ADP and phosphate. And the second role of the calcium ions is to essentially move tropomyosin out of the way to allow the binding sites on the thin actin filament to then be exposed, which would allow the myosin head to bind to those. Right, the last point then at the bottom of our activity, the energy released from the hydrolysis of ATP recocks the myosin head. That goes back into an active position where it can then form a cross bridge, and that movement of the myosin head into the active position is called the recovery stroke. So all these red terms on this activity you guys will have to learn, okay, and be able to write about as, for example, a 5 or 6 mark exam question.